Our speaker today is Sara Leek, and she was actually she she was born and raised in St. Petersburg, Florida, which might indicate the reason why she leans towards biology, but I'll let her talk about that. She moved to Plainview, Texas, the last 12 years, and until she moved to the North Texas area to work on her master's at UNT in the fall of 2019. Today she's going to present, well, first of all, she has a BS in biology from Wayland Baptist University, and she is going to present today determining home range movements and habitat use for a fragmented population of three-toed box turtles in North America. She's been doing a lot of research at LLELA, and many of us in the Elm Fork chapter have been volunteering and assisting her in her research. One of the things that we're doing is we've been raising money for more transmitters for the turtles, and I just want to show you the t-shirt, which says, I'm a lover of box turtles. I'm a box turtle lover. So with that, Sara, I'll turn it over to you.
Okay. So thank you for the introduction, Rita. I'm going to be talking about the LLELA Box Turtle Project. And the title I gave Rita that she just so kindly read to us is the title of what my master's thesis is going to be. But I'm going to be talking a little bit about some other box turtle stuff going on at LLELA as well that I'm helping with. So Rita just said a little bit about me. I lived in Plainview, Texas for 12 years and went to Wayland Baptist University. I did an undergraduate thesis on Texas horned lizards. So that's kind of where my love for biology started. My parents are both veterinarians, so I grew up in and out of vet clinics, and I worked at one for 5 years, and I thought that's what I wanted to do with my life until I started doing research on horned lizards. And now I'm here doing my master's on box turtles. So it's a little different than veterinary stuff, but hey, it's still animals. A little bit about three-toed box turtles. So there's actually 4 different subspecies of box turtle in the US. The common box turtle is just known as Terrapene carolina. The three-toed box turtle subspecies is Terrapene carolina triunguis. The IUCN does not have any listing status for the subspecies of box turtles. They just have it for the common box turtle in general, and they're listed as vulnerable. In Texas, the three-toed box turtle specifically is considered a species of greatest conservation need. And they've been declining pretty much all over, mainly because of the illegal pet trade, habitat loss, and road mortality. It's always really sad when you see little turtles hit on the side of the road. So there's a lot of variation in their lifespan when you're looking at the literature. They can live like 30 to 100 years old, just depending. They have an incredibly low reproductive output, which has also contributed to their decline. They typically only lay like 1 to 2 clutches of eggs per year, and each clutch only has 2 to 5 eggs. And unfortunately, a lot of these clutches end up being eaten by predators like raccoons or coyotes. Box turtles are omnivorous. They'll pretty much eat anything that looks good and will fit in their mouth, including stuff like roadkill, which most people probably don't think a box turtle would eat, but apparently they will. I think they're really cute, so I had to throw that in there. And box turtles are called box turtles because their shell basically makes a box. Like if we're looking at like a red-eared slider, which is like an aquatic turtle, they can't close their plastron. But box turtles can completely close their shell and keep all their extremities inside, which gives them more protection from predation. There's been debate that I've read in the literature about whether or not you can age box turtles using their scoot rings. So Each of these, I guess, kind of plate looking things is a scoot. And then all these little rings are growth rings. So some literature's reported that turtles only put on one annual growth ring, but there's other literatures that has reported they can put on like 2 to 3 growth rings a year. So it's really not a very accurate way of aging them. And as far as we know, there's really no way to get an accurate age estimate once they're in kind of like the adult age class. And then I figured somebody would probably ask me about how you sex them, so I thought I'd go ahead and talk about it now. So male box turtles have a concave plastron to allow them to mount the females. So that's one way we can tell. Male box turtles also have like a redder eye color than females. Females have brown eyes. So those are two of the ways that you can kind of tell the differences in the sexes. Unfortunately, there is a lot of limited knowledge in Texas. So in all my literature searching, most of the literature that's been published on three-toed box turtles is in Missouri or Illinois. And that's definitely a long way away from Texas. So there's just really not information on the home range size that these box turtles need in Texas, their habitat use, their reproduction, or like their population size as a whole. So given that, our research goals are to, 1, determine home range movements, microhabitat associations, and habitat use for resident three-toed box turtles at LLELA. And our second goal is to determine if head starting is a viable conservation tool for three-toed box turtle populations. The first goal is what I'm focusing on for my master's thesis, but I'm doing a lot to help out with the second goal as well. And I'm hoping that it can possibly become part of a PhD, because I do want to continue school after I'm done with my master's. Okay, I was going to give a little bit of a rundown on LLELA. I know most of you probably know what LLELA is, but it's the Lewisville Lake Environmental Learning Area. Their mission is to preserve and restore native Texas ecosystems and biodiversity while providing opportunities for environmental education, research, and recreation. So LLELA is south of the Lewisville Lake Dam. In 1955, the land that's now LLELA was purchased by the federal government from homeowners and farmers to kind of give a buffer zone below the dam. And then in the 1990s, the organization we know as LLELA began. LLELA currently has numerous ongoing research projects, habitat restoration projects, and reintroduction projects that are ongoing. It's a lot of cool stuff that's going on. I'm not going to try to list it all because I'll probably leave stuff out, but it's definitely really cool that LLELA is a place that a bunch of like students from UNT and people from like Master Naturalist volunteers can go out and learn and help with all of this research. And I did want to show you this because box turtles are so long-lived. We know that all the wild box turtles that are currently in my study are probably about over 10 to 20 years old. Like I said, you can't really get a great estimate of age, but if we had to guess, that's probably about how old they are. So this is a National Land Cover Database map of the land cover at LLELA in 1992. And you can see all the different colors, and the key is over here on this side. So in 1992, a bunch of the land cover was pasture and hay, as well as cultivated crops, which obviously probably is not the best habitat for box turtles. They're kind of slow. I somehow doubt they'd be able to get out of the way of any tractors or anything like that. But now this is LLELA today. So we have an entire different scenario. LLELA itself is now mainly deciduous forest and grasslands, but all of this pink to red coloration is urbanization. So it's all developed. LLELA's basically become kind of this habitat island, and it would be incredibly difficult for any of these box turtles to disperse elsewhere. So that could have potential implications on the population genetic structure and that sort of thing, which I haven't looked into yet, but if I do a PhD, that's hopefully where I'm going to go with it. But for now, just know it's really fragmented. And you might think that maybe the box turtles could travel south this way, but there is a 4-lane highway right here that they'd probably have to cross to get south. So there's really nowhere for them to disperse. So a little bit more of specifics. I'm trying to simplify it by calling the properties by east and west side. So the whole thing is LLELA. We've ended up with 2 separate kind of groups of box turtles that we're tracking, just because that's where we were able to find box turtles. So we have a group that we track on the west side of the property. The west side of the property is, if you know LLELA, up by the offices. The habitat there is all cross timbers habitat. And just visually, it has a lot less understory than the habitat on the east side of the property, which is bottomland hardwood forest. So in my research, I'm going to be trying to make some comparisons between the different habitat types, and I'll be referring to them as west and east, just for future knowledge. So moving on to some of my methods. First, we wanted a way to be able to identify all of the individual turtles that we're marking or putting transmitters on. So to do this, I'm using a method called shell notching. And the code system I'm using is called the North American code. Because as you can see here, on the left side, it starts with N, and on the right side, it starts with A. So the right side starts with A and goes clockwise to L, and the left side starts with N and goes counterclockwise to Y. And this NA is why it's called the North America coding system. This example, this turtle would be, the code would be AOXY. And then this is an example of what it actually looks like, the shell notches on a wild turtle. And so for example, this is AB. I'm sure I have another notch somewhere on his shell, but in this picture you can just see AB. And it does get a little difficult because a lot of the turtles do have different scarring and I guess deformities almost in their shell. So you have to be really careful to make sure you're looking at actual notches and not other damage. So 2 to 3 times a year, morphological measurements of all of our turtles that we have radio transmitters on. They don't really enjoy it. We take a weight, and we also take a bunch of different measurements of their shell. This girl was definitely not happy about that day. She watched the entire time. I was like, what is going on? But it's a lot of fun being able to handle them. So I use radio telemetry to actually locate these turtles. So in order to get a transmitter, which is this little gray thing, put on a turtle, we have to find the turtle in the wild, which can be difficult. Most of the time, it's just kind of like random encounters where we accidentally stumble upon a new turtle and are like, yes, we found a turtle. But sometimes we find them mating with previously marked turtles. So that's helpful. And I wanted to talk a little bit about how radio telemetry worked. So kind of what is radio telemetry? So each box turtle is fitted with a radio transmitter, and each transmitter emits its own frequency. And I'll show you kind of a picture of what that looks like in a second. So the frequency it emits is then picked up by a Yagi antenna. And we use a 3-prong Yagi antenna, and then that's connected to our receiver, and the receiver kind of interprets all of the information that the antenna picks up. So this is a picture of the receiver and kind of what it puts out. So I said each turtle has its own frequency. So this turtle's frequency was 166.996. So this turtle, I know that frequency. It's actually our turtle M7, and his name's Don Juan. And then this is the signal strength. So it basically tells you how close to the turtle you are. So the higher the signal strength, the closest you are. It only goes up to about 200. So once the signal strength hits 200, you're basically right on top of the turtle. And the gain kind of adjusts the area that the Yagi antenna is like searching for a signal. I'm not going to get into that because it's really confusing to explain. It makes sense when you're out doing it in the field, but I don't want to confuse anyone, so I'll just leave it at that. So how many turtles am I actually tracking? It actually changes all the time. Right now we have 8 turtles on the west side and 6 turtles on the east side. It changes anytime a turtle loses a transmitter or anytime we find a new turtle and we have a replacement transmitter. Unfortunately, the research has not been without complications. As you can see here with these 2 transmitters, I found the transmitter and no turtle, and both the transmitter had been chewed on. They had very obvious bite marks. So we're trying to figure out if the turtles that these transmitters were on actually survived. Like, was an animal just kind of chewing on them and messing with them? And did their shell do its job? And the transmitters just got damaged, or was the turtle actually killed in the process? Fortunately, right now, we don't know. If we ever manage to recapture any of the individuals that lost transmitters at some point, I guess we'll be able to figure that out, but time will tell. Another thing was, at one point, we accidentally broke the Yagi antenna, so that was a mini disaster. Thankfully, we were able to fix it, but it's the kind of stuff that happens in field research, so it's how it goes. So once we use the antenna and the receiver to locate a turtle, we always try to locate them visually, make sure we can see them, make sure the transmitter is still on. And then once we've done that, we'll take a GPS waypoint at their location. And then we'll take microhabitat measurements at their location as well. And yes, somebody pointed out that's Barbara Bean. Yes, that is Barb. She is very helpful. I thought that picture was really funny. There's a box turtle somewhere in behind her, but the poison ivy is as tall as she is. So it's another thing that we have to deal with a lot when we're out in the field is, do we really want to crawl through all that poison ivy? Sometimes we do, sometimes we don't, depending on how close the turtle is and if we can kind of estimate their location. So for the microhabitat measurements I'm taking, I'm recording like percent substrate type. So if it's litter, grass, forbs, or bare ground, the percent understory cover, as well as the percent canopy cover. And I'm doing all of that in a meter squared centered on the turtle. I'm also recording a percentage shell cover. So like this turtle has like less than 5% shell cover. And then on top of that, at each turtle location, I'm also recording the air temperature, the relative humidity, and the surface temperature of the ground. Once we've recorded all of that at the turtle's location, I'm also taking pretty much the same measurements at a randomly selected location. That way we can compare and see if the turtle is selecting specific habitat. So to get my random location, I have a random number generator that generates 1 through 4. 1 is north, 4 would be west. So then once I have my random direction, I head off 5 meters and take the same measurements. We decided on 5 meters because we can assume that that is an area where the turtle can get to. So it's habitat that it could be selecting and it's not selecting. So hopefully we'll end up getting some good data from that. On top of my microhabitat analysis, I'm also planning on doing some habitat analysis in ArcGIS, which is a mapping software. Like I showed earlier, this is just a little snippet of the National Land Cover Database map. So I could use this to estimate like percentage habitat type in their home ranges, but I've I'm going to try to find a map with a higher resolution showing the kind of smaller fluctuations in habitat type. If I can't find one, I'll end up having to make one myself, but I should be able to do that. I know there's some GIS professors at UNT that know how to do that process. It's not something I've started yet, but it is in the works. And then it was actually for a class assignment. I did normalize difference vegetation indexes for our turtles in 2019. And I'm going to talk about that a little bit today. For now, just know it's the NDVI. The highest values of 1 mean like the most photosynthesis is occurring, and -1, or like the white areas, mean no photosynthesis is occurring. So it's kind of a proxy of like how much food availability there could possibly be based on the amount of plants and amount of photosynthesis. So I am trying to look at the reproduction of the wild turtles on the property. This year it didn't go so great. We collected all 6 females that we had transmitters on and transported them to a veterinary clinic for x-rays. Some of them were not happy about that. And then this is the x-ray they took, and unfortunately none of the females were gravid. We did this in kind of like end of May, which is when they should be gravid and hopefully about to lay eggs. I'm not sure why they weren't gravid. It's possible that I missed it and they laid eggs earlier, or maybe they were going to lay later this year, depending on environmental factors. I really have no idea. So next year, I'm hoping to x-ray all the females in April, May, and June to see if we can catch any gravidity at all. Okay. So now I'm going to show some incredibly preliminary results. It's all really preliminary. No analyses have been completed yet. Technically, I have not even done my thesis proposal for UNT at this point. I've mainly been in the field doing stuff, so I haven't even gotten my thesis proposal approved. But keep all that in mind. So for 2019, I already have, some maps made. So this map is a comparison of 2018 and 2019 for 2 turtles that we had enough GPS locations for Fangs and Gypsy. So Fangs is over here on the left side in the orange and purple, and Gypsy is on the right with the blue and green. And it's pretty interesting because there's definitely like a stark difference in their home range size between 2018 and 2019. Part of this might be because the turtles were only tracked closer to like midsummer to the end of 2018, because that's when they were first captured and had their transmitters put on. And then they were tracked like all summer in 2019. So I'll be interested to see, we still have both of these turtles that we're tracking, so we can see what their home range is going to be this summer and see how different it is compared to these. So with that NVDI that I showed you guys earlier, the black and white map, I did like a estimate of vegetation cover. And for Gypsy and Fangs, it doesn't really look like there was a difference in that vegetation cover between 2018 and 2019. So this is just a pretty map of all the turtles that we tracked in 2019 that had enough GPS points. I guess one thing to point out here would be, like, all of these turtles seem to have, a pretty similar home range size. And then there's Gypsy, who is just all over the place. There is some stuff in the literature that says turtles can be transient. So it's possible that Gypsy is transient and just kind of wandering around doing her own thing and hasn't settled down to form a home range. Either that, or if the habitat is different enough there, maybe she needs to use that much more home range to fulfill like her life requirements, like food and water and shelter. So I'm really interested to see what this is going to look like for this year. And then this is over on the east side of LLELA, so in the bottomland hardwood forest. Once again, everybody's pretty much the same size. Don Juan didn't have very many points because we just captured him the end of 2019. KB is interesting. I'm wondering if she's transient as well. But her data from 2020 will hopefully show us. Unfortunately, she lost her transmitter last week, so we're only going to have part of 2020 for her, but I'll see what I can do with that. So I compared male and female home range area and vegetation cover. And once again, I haven't done any statistical analysis, so it looks like there could potentially be a difference in male and female home range size. However, this could definitely be skewed by Gypsy's giant home range size. So I definitely need to do more analysis on that. And then it does seem like vegetation cover is similar for both male and female home ranges. So then I also compared the east and west side. So the bottomland hardwood forest and the cross timbers forest. The home range area looks similar. You can tell this is standard error. So there's a lot of variation in the home range sizes on the west side. And that's once again, probably because of Gypsy, because she's on the west side of the property. And then for the west side, or the comparison of east and west side vegetation cover, I didn't do any statistical analyses, but looking at like the standard error bars, it does look like this could potentially be significant, but we will see. I'm hoping to do this again for this year as well, and see if there's any differences. So discussion and conclusions for the little bit of stuff I have done for 2019. There was a fairly similar NVDI and home range area for all of our box turtles, except for Gypsy. So Gypsy's probably transient and is an outlier in my data set. All of this was done with really low sample sizes, so we need a ton more data, which I'm working on. It's a slow process. So for 2020, I went ahead and put some stuff together just from data that I've collected pretty much like April through June. So it's not going to be my full data set, but I thought I should go ahead and show some of it. So once again, I haven't done any analysis. So this is a comparison between our turtle locations and the random locations, like how I described earlier. The turtle locations are in blue and the random locations are in orange. This specific bar chart is for this percent substrate within that 1 meter squared area. And for the most part, it does seem like it's a pretty similar percentage of substrate cover at turtle locations and random locations. However, there does seem to be a fairly large difference in the percent understory cover at turtle locations and random locations. So that could potentially mean that turtles are selecting for a higher percent understory cover, potentially for thermoregulation or protection from predation. The canopy cover at turtle locations is slightly higher, but I don't think that would be significant. And once again, I mean, we're going to be collecting a lot more data on all of this, so hopefully any differences will continue to become more obvious as the project goes on. So I did those same comparisons we just looked at for turtle locations and random locations with male and female locations. Once again, a lot of this seems pretty similar. Females seem like they have a little bit less litter cover and a little bit more bare, like bare ground. Potentially that could maybe be because they're looking for nest sites, but I'm not sure that's really a big enough difference for it to be significant. Once again, I need more data. You'll hear me say that a lot. And for understory cover and canopy cover, it does seem like males and females are using a similar amount. which makes sense, especially if they're using it for thermoregulation and for protection from predation. I don't really see why there would be a difference in males and females. So I also did these comparisons with the east and west side, so the cross numbers habitat and the bottomland hardwood habitat. And it does seem like there could be some potential differences once I have more data in these different habitat types. I don't want to say anything concrete yet. And it definitely does seem like potentially understory cover will be significant between the west and east side. Time will tell, but canopy cover is similar. So discussion and conclusions on everything I've done in 2020 so far. I need a larger sample size. I need a lot more data. I need to perform analyses, but already there's some trends that seem to be appearing. So it should be really interesting to look at this full data set whenever I'm done collecting everything. Okay, so the second goal of the project, that was all for the first goal. So that was like all what I'm doing for my master's thesis. The second goal of the project is head starting. So head starting is the process of raising captive animals through the more delicate life stages and releasing them when they're more equipped to survive. So like for our little box turtles, they cannot completely close their shell until they're like over 100 millimeters in carapace length. So I think that's probably about 3 to 4 years old. So we're raising the turtles for our head started study until they're old enough to close their plastron. And then previous research on other box turtles and on three-toed box turtles in Missouri showed that a larger body size typically equates to a higher survival probability. So we wanted to raise these guys to the point where they'd have a much higher survival likelihood. So how do we care for all of them? So we have a really amazing team of volunteers. They go out 3 or more times a week to check on all of our little box turtles. We currently have 24 total. They're fed a mix of greens, veggies, fruits, and insects. They also receive calcium powder 3 times a week and a multivitamin once a week, just to make sure they're getting all the nutrients they need to grow. And then I have some pictures of the enclosures, but the enclosures were also designed to offer a gradient of sunlight so the turtles can thermoregulate as needed and get the vitamin D3 they need. So these are some of the enclosures, or 2 of the enclosures that are on the west side of the property near the offices. And then these are the enclosures on the east side of the property. As you can see, they're pretty sturdy. They were predator proofed incredibly well because we didn't want anything to get in there and try to eat them. And so far we've had success with that, knock on wood. So our future plans for head starting, because it's definitely started because we've been taking care of all the head started turtles, but we haven't released any yet. We have 10 3 to 4 year old juveniles that we're planning on releasing this fall. And they're all going to be equipped with transmitters. So I'll be tracking them as well. And then we have 14, we're estimating they're about 2 years old, that we're going to let grow for at least another year, possibly 2, just so that they're large enough to close their plastrons and large enough for the radio transmitters we're using, because we are trying to keep the weight of the radio transmitter at less than 5% of the turtle's body weight, just so it's not interfering with them or their movement. So future plans, we're going to do a lot more tracking, especially of our wild resident turtles until the end of fall 2021, hopefully. We'll be reintroducing some of our head started turtles this September, so that should be exciting. I'm hoping to continue tracking any of the juveniles we release for the next 3 to 4 years, if they'll keep their transmitters on, which is always a fun thing to guess about. And then next year, I'm going to try to x-ray all the females again and see if we can figure anything out about that. Mainly because there's so many activities that go on at LLELA, and nobody's ever actually seen baby box turtles at LLELA. So we're not sure where, like, in the reproduction cycle we're losing babies. It's probably because of predators eating nests, but we don't know that for sure. So I wanted to try to examine the process and see if I could figure out how we can help the wild population a little bit more. So acknowledgments, Friends of LLELA has funded this entire project so far. Scott was the original volunteer that was doing all of this, Scott Keister, so he taught me everything. And then all the other volunteers that we have come out, like Barb, and then I used to be able to have UNT students come out with me. Now with COVID, I haven't been able to do that. But all the help we can get is absolutely amazing. And we're always very thankful for it. And then I have a little question slide, but I'm gonna skip this for a second because this is a contact us page. So I have a Facebook page, the LLELA Box Turtle Project Public, where I post updates. So if you guys want to see any of that, you're welcome to check it out. We are selling t-shirts like Rita mentioned, so there's a link to that as well. If you want to learn more about LLELA, I linked the LLELA website. If any of you are interested in getting more information from me or would like to volunteer or anything like that, my email and my cell number are there at the bottom. So with that, questions?
Well, we do have some questions for you, Sara.
I know, I see that.
How long do the transmitters last, and what's the transmitter's range?
So the transmitters last depending on how large they are. So the larger they are, the larger battery they can have. Right now we have, I think pretty much all we have in rotation are 15-gram transmitters, and those last for 2 years. They have a 2-year battery life about. And then there's 10-gram transmitters, and those only last about a year. And the transmitters I'm putting on the head started turtles are 6 grams, and those only last 6 months. So there's a lot of variation with that. So then the range, that's a good question. I'm still trying to figure that out. Because sometimes it seems like you can pick up a signal from like a pretty good distance away, and then sometimes for some reason you don't pick it up. It does seem like any like changes in the topography or like really thick, dense vegetation kind of interrupt the signal somehow. We've definitely kind of walked around in circles before looking for turtles, so it's definitely a learning process.
And so did you name Gypsy before or after you knew that she was a traveler?
I think after. Scott and his volunteers named her before I came along, and they realized that she kind of went all over the place, so she got the name Gypsy.
How many turtles do you estimate total, I guess, in LLELA?
Right now, we've only had our hands on 20, so we know there's at least 20 turtles. I was trying to train a dog to track the turtles and like find ones that we don't know about, but that ended up just being too time consuming, especially considering everything else I was doing. Really, they're so cryptic and so hard to find, it's difficult to know. But I know reading some of the literature from like the 1950s, it definitely seems like our density of turtles is a lot lower than back then. Because like one of the papers I read about like their hibernation said that you could find like turtles piled on top of each other in the areas that they like to hibernate. And we definitely haven't seen that. So I feel like the population density is probably a little low.
Let's see, Cicely wants to know, do you think that the estimated age of the turtles makes a difference with their preferred range?
Yeah, I think definitely, which is why it's kind of a bummer there's not a better way to age the turtles. We pretty much just have to either say like juvenile or adult. But I think, like, especially depending on environmental conditions as well, because female box turtles can actually store sperm. So they can mate and then not lay eggs until a couple years later. So there's so many different things that probably affect their home range size. And I would imagine there's a bunch of Like Gypsy, for example, she could have been traveling that far just to find somewhere to lay eggs. I'm not sure, but that's definitely a possibility. I feel like there's just so much we don't know. So I hope I answered that all right.
Yeah. Abigail wants to know, where did you get the little ones?
Okay, so all of our little guys were donated by Dr. Carl Franklin. He's a herpetologist. He runs the Texas Turtles webpage and Facebook page. He's really into turtles and knows a lot about them, and he donated the turtles to the project.
Right. Will they all be released in LLELA, or do you have other areas in mind as well?
Currently, they're all just going to be released at LLELA. When I start thinking more about a PhD, I might try to find some other areas to maybe release some, but that's all very kind of up in the air right now.
Okay. Are there any other ongoing programs like this in the state?
Not that I know of in Texas. There is a program that's Head Starting Turtles in Missouri. And I've emailed with the lady in charge of that a little bit. But as far as I know, in Texas, there's not.
Mario wants to know, does transmitter interfere with mating physically?
No, we do try to put the transmitters, let me see if I can find a good picture. Okay, so we try to put them kind of to the back and to the side so that they don't interfere with any mating going on. And there's actually a quote from the box turtle book. I don't remember what the title is, but the author says pretty much nothing can stop a male turtle from mating when he is trying to. So we've seen the mating. I don't think it's interfering with anything. Apparently they're kind of unstoppable where that's concerned.
Irene wanted to know if the juveniles turtles were from the area. So the doctor you got him from, is he local?
Yeah, I think they're all pretty much from around North Texas.
Jeff wants to know, if we were on the property and spot a turtle, how can we report that information to you?
Okay, well, one, you're definitely welcome to like call or text me. I'm out at LLELA either like 2 to 4 days a week, and if you find a box turtle, I will probably come to you and put a transmitter on it. And you can also post it on iNaturalist. Oh, that was another thing I should have linked that I forgot about. So I also have an ongoing like iNaturalist project just for anybody at LLELA. If you see a box turtle, please like take a picture of it and post it. That way we kind of know where any that we haven't marked are, and to kind of try to get a better estimate of the population. But for right now, I do have transmitters I need to use. So if y'all find a box turtle, please call me.
Okay. How old are the females when they start laying eggs?
I think about like 8 to 10 years old. But there's some variation in that as well.
Susan wants to know, is it true? Whoops, is it true that captured relocated turtles will try to return to their original location?
Yeah, so there's been a bunch of studies on translocation of box turtles, and it's all had like really mixed results. Like some studies were pretty successful in translocating box turtles, and the box turtles stayed around and established a home range. And then other studies, they tried to go right back to where they were from. So it probably depends on some like environmental variables or the turtle's own kind of personality, because they definitely all seem like they have their own personalities. It's just hard to say, but typically it's better. Like I know a lot of people when they find them on the road or whatever want to take them home or something where they're safe, but it really is better to just move them to the other side of the road and let them continue on their way.
Good to know. What do the letters stand for other than North America?
Yeah, so the NA is just North America. The other letters are just so we can use that code. So each turtle will have like its own individual code. But For all of the like adult turtles right now, I'm doing 3 notches. So they all have like a 3-letter code. So like ABH or ABJ. And I'm going to go through and do like all the 3-letter codes I can before I move on to 4-letter codes. If that helps explain it a little bit.
If box turtles are found on our personal property, is that information you would want to be informed about?
I don't know. It's really cool that they're on their property. Unfortunately, right now, I don't think I really have time to do anything with that kind of information. At some point, I may or may not be looking for like a larger property to do head started studies on, depending on how all this goes. But that's up in the air as well right now. So,
what's something they could put on iNat?
Yeah, they could definitely put it on iNaturalist, and then it's there if either me or anybody else would need it.
Yeah. Okay. Rodrigo, is it known how long a turtle typically remains transient before it establishes a home range?
I don't know if the studies that I read mentioned that specifically. It would probably depend on the turtle. There's just so much we don't know about, like why they wouldn't establish a home range and would try to go back to where they're from and that sort of thing. And like sometimes a lot of turtles will suffer like a mortality before even like establishing a home range when they're translocated. So it's difficult to study.
Yeah. We have any other questions?
Well, let's see. I think Michelle had one. Has any genetic testing been done? Do we know if the population is diverse? So no genetic testing has been done on our population. I was actually emailing with somebody the other day who's doing, I guess, kind of like a nationwide genetic study to create a database of, I guess, genetic information so that when turtles are confiscated from like, I guess, the black market, basically, they can be returned to close to their like original locality based on genetics. So that's pretty interesting. I didn't know until recently that that was going on. I do plan on trying to do some genetic work on this population, but we'll see. That'd be a few years away.
Let's see, we have some more here. The notches are not natural. How do you put them on?
Okay, yeah, so they're definitely not natural. Sorry, I forgot to mention that when I was on this slide. I have a little file, and you just file it into their shell.
Can we make a donation to Friends of LLELA dedicated to the Turtle Project?
Definitely. So if you go to the Friends of LLELA link I had at the end of the slideshow, which, there should be a donation button on LLELA's website, and you can donate through that. And just make sure to add a comment that it's for the Box Turtle Project specifically.
Diane Weatherby has a note saying, interesting research project would be to see how box turtles are adapting to developed areas. Here in Lantana, just south of Denton, we seem to have a lot of box turtles. Although highly developed, the riparian areas along the creeks have been maintained as natural areas. Wondering if that contributes to maintaining our population.
Yeah, I mean, I don't know in box turtles, so that's really been researched a whole lot, but I know like little corridors like that can really help with gene flow and help maintain like a better like population genetic structure. So it probably is really helpful to have those little areas where they can live, because that'll help connect some of the bigger populations that are further apart too, hopefully.
All right, any more questions for Sara? Thank you, Sara.
Yeah, definitely, you're welcome.
It's been very interesting, and we hope to have you back to learn more of what you found.
Yeah, definitely.
Great, thank you.
Yes, thank you for having me.